Hi, welcome to Microbiology Mentor. Today we will be discussing about another important topic, laboratory diagnosis of malaria. So malaria is a protozoal disease which is transmitted by the bite of female Anopheles mosquito, and it is caused by not less than 5 species of plasmodium like falciparum, vivax, ovale, malari, and recently the knowlesi. Malaria is endemic throughout the tropic and subtropics. Let's see the diagnostic options available for malaria, which includes microscopic techniques like making a thick and thin smear from peripheral blood examination, quantitative buffy coat, fluorescence microscopy, as well as non-microscopic techniques like antigen detection test, culture, and molecular methods. So let's see what should be the blood sample, and ideally a blood sample obtained either by pricking a finger or earlobe is a better sample because it has trophozoites and schizoid in a greater concentration, because that area is peripheral, which is capillary rich area. Then the blood obtained either by venipuncture, which is collected in heparin or EDTA, is although an acceptable but an inferior specimen, because morphology of WBC and parasites alter after a prolonged storage in that, which may hinder in the diagnosis while you perform a test using microscopic technique. So let's see the peripheral blood smear. So the peripheral blood smear was considered as a gold standard, and even now it is considered a gold standard. So thick smear first, which is made by concentrating the layer of RBC on a small surface. After that, the RBC are lysed using a hypotonic saline or tap water, which is then further stained by using Giemsa, Leishman, Fields Wright, or Jasbant Singh Bhattacharjee stain. This method is comparatively more sensitive because it can detect as less as 5 to 10 parasites per microliter of the blood. Whereas thin smear, which is prepared by making a blood drop spread on a slide, which gives a uniform layer of the RBC, which is again methanol fixed, stained with Giemsa or Wright, and which helps to visualize the parasite in the RBC. Morphological identification of the parasite even to species level is easily done using a thin smear peripheral blood examination. It is assumed to have a greater specificity than thick film because the morphology of the parasite is easily visualized. It is preferred for routine estimation of parasitemia because organisms are easier to see and count. At the same time, it is helpful in monitoring the response to a therapy because sequential count of the parasite in the blood film can be done. Now, diagnosis using fluorescence microscopy, which is easily done by staining the slide with a fluorescence dye like acridine orange or benzothiocarboxypurine, that is BCP, which have an affinity for the nucleic acid of parasitic nucleus. After that, the slide is excited by a UV light at an appropriate length, which visualize the nucleus, giving a strong fluorescence, which appears as apple green or yellow fluorescence. A Acridine-orange is used either as a direct staining technique or sometimes this can be combined with a concentrated sun method such as thick blood film. Now, quantitative buffy coat. So, in this centrifugal quantitative buffy coat combines acridine-orange coated capillary tubes with a centrifugal force to separate the layer of RBC and platelets. The parasite concentrates appearing in the upper layer of RBC. The parasite can be visualized through capillary tube using a special long fluorescence length objective and a fluorescence microscope. Now coming to the non-microscopic techniques, in which first is antigen detection test. So based on the principle of immunochromatographic antigen capture, which are capable of detecting 100 parasite per microliter of parasite, giving as rapid as results in 15 to 20 minutes. So commercially, the test using an antigen detection is available, and it has an advantage that it does not require any extensive training or equipments to perform or to interpret the results. The example of antigen detection test includes Plasmodium falciparum HRP2, PLDH and P. eldolase. All these antigen detection tests are available in ICT format commercially. Let's see the histidine-rich protein test. So this test is specific for Plasmodium falciparum because the infected RBC synthesizes at least 3 histidine-rich protein HRP1, 2 and 3, out of which HRP2 is a water-soluble protein which is produced by asexual stages and young gametocytes of Plasmodium falciparum. This is expressed on RBC's membrane surface. Regarding the other targets of antigen like Plasmodium LDH as well as eldolase. So first PLDH, which is found in the glycolytic pathway of malarial parasite, which is produced by sexual and asexual stages of the parasite. In this also different isomers of PLDH are available which infect human egg parasites. So several other enzymes of malaria parasite like eldolase are also used as a target for antigen in a rapid detection technique. Now regarding antibody detection. So antibody can be detected using ELISA, immunofluorescence assay, or rapid detection tool formats available using soluble malaria antigen. However, the detection is controversial in certain situations because antibody persists even after the clinical cure. So this technique of antibody detection does not differentiate between the current and past infection. So in endemic zone like India, Government of India has banned the use of antibody detection test for malaria detection. Now regarding culture. So culture is usually not of diagnostic use, but yes, it can be used for preparation of antigen. So Traeger and Jensen discovered the simple method for continuous culture of Plasmodium falciparum, which was later extended to other species of malaria using RPMI 1640 as a liquid culture media. However, later there were other modifications like Dalbecko's modified eagle method using RPMI 1630 and medium 199. Now regarding molecular methods, PCR assays have been developed for detection of malaria DNA from whole blood. It has a strength in its ability to detect approximately as low as 5 parasites or even less per microliter of the blood. At the same time, it can be used as a tool in certain difficult situations where the morphological problems arise due to attempts to identify parasite to species level. With this, I finish my talk. Thank you.